So Alex Reif is a senior staff researcher at InstaDeep, currently focused on applications of generative AI to biology. He was previously chief scientist at NNAISENSE and a senior researcher at Google DeepMind. Prior to that, he earned a bachelor in theoretical physics from the University of Edinburgh and a PhD in artificial intelligence under Jürgen Schmidhuber at IDSIA. He was also a postdoc under Geoffrey Hinton at the University of Toronto. His contributions include the Connectionist Temporal Classification, RNN transducer algorithm for sequence labeling, both widely used for commercial speech and handwritten recognition. Stochastic gradient variational inference, the Neural Turing Machine, Differentiable Neural Computer architectures, and more recently, Bayesian Flow Networks. So with that, Alex, it's up to you.
Which microphone? Oh,
you can this one. You can maybe take those ones for questions later.
Transition to full screen, and then you're ready to go. Sorry.
Lovely. Okay, thank you very much, and thank you for inviting me to this winter school. Okay, so I'm going to be talking about AI for biology, and how we can take some of the these exciting developments in generative AI that have happened over the last few years, and start using them to drive kind of discovery and innovation in scientific fields such as biology. Okay, so I'm going to begin with a very basic recap here. So I'm going to be talking, and I'm sure this is familiar to most of the people in the audience, but it's always worth kind of like refreshing these basic ideas. So I'm going to be talking a lot about joint and conditional probability distributions, and just to kind of recap what I mean by that. So let's say we've got a simple system here with two variables, x and y. The joint probability distribution of x and y is shown by this kind of hump in the middle of the picture, where the height of the hump tells you what the probability density is at each point. So it's kind of telling you how likely are certain values of x and y to co-occur. And obviously, because it's only two dimensions, it's quite simple. We can plot the whole thing. We can sort of analytically, we can analyze the whole thing. And then there's like three other kind of distributions that I want you to think about here. One of them is there's a pair of marginal distributions. So the probability of x and the probability of y independent of one another. And these are the two kind of Gaussian distributions that you see plotted along the x and y axis. I like to kind of think of these as sort of like the shadow. This is like the shadow of the distribution cast onto a particular axis. So if you sort of flatten everything along the other axis, you're left with this trace. And what it's kind of allowing you to ask is, okay, irrespective of what y is, how does x behave? And then the other really important distribution is the conditional distribution. So this kind of shaded slice here through the diagram is saying, well, what if I fix a particular value of y in this case? What if I say y equals d, then what does the distribution over x look like? And very, very basic mathematics, but this is something that we can leverage to start asking really interesting questions. Okay, so the first key point to make here is that that slide I just showed you is a distribution over two variables. The whole reason that generative AI is so exciting and the whole reason that it was so difficult to make it work is that it involves distributions over millions of variables. So in this case here, we're talking about all of the pixels, actually probably all of the red, green, and blue channels in all of the pixels in a high resolution image of a person's face. And this is a joint distribution over millions of variables. It's way beyond anything we could have done with conventional statistics. There's no way to even sort of write down an explicit distribution over that many variables. But what you can do is learn a distribution, implicitly learn a distribution like that inside a deep neural network. And that's exactly what the whole kind of generative AI revolution is about. And I say it's implicit. Basically what I mean is you can't write down an explicit form for the distribution, but you can draw samples from it. So it's always about drawing samples. That's what we're always doing with these generative models. And just to give a kind of simple illustration of the difference between joint modeling and kind of marginal modeling. So, you know, obviously if you were to say, what's the marginal distribution of any one pixel in an image, then, and treat those as if they were all independent and draw a sample from that, you would just get an image like the one on the left here. It's just pure noise, right? There's basically no information in the distribution of an individual pixel. All of the important information is contained in the relationships between the pixels. And it's just kind of, it's worth stopping and thinking just how incredible it is that these models can learn that, right? In order to generate a face like that, they're having to learn so much, you know, implicit kind of structure about the human face, that it's roughly symmetrical, that the color of one eye generally matches the color of the other, that skin and hair remain sort of constant, or not constant, but at least they're kind of like, you know, continuous in texture and so forth. All of those things are somehow encoded in this neural network, and they're a part of this joint distribution. But it's the key point I really want to get across is it's all about relationships among variables. That's what makes these models powerful. Okay, so in the previous slide, it was kind of like, that was the joint distribution, right? That's what, sort of like an unconditional sample from the joint over all the pixels. Usually we want something a little bit more nuanced than that. What we actually want are conditional distributions, and we want conditional distributions because we're going to use the conditioning to guide what the model does. So here's a pair of generative models that were both released in 2021 by OpenAI, DALL-E and CLIP. Very famous models, but what's kind of interesting about them is that they're basically inverses of each other, right? One of them is essentially modeling the conditional distribution of images given captions, and the other one is doing the exact opposite. What's the distribution of captions given images? But you can sort of, you know, you can see if you think about it, they're both accessing the same underlying joint distribution, the joint distribution of images and captions. And so if you imagine if you had access to that, a single model with access to that distribution, such that you could sample, you know, either of these conditional structures, you'd kind of have, you'd have two models in one. So the point I'm trying to get across here is that, and this is something that's kind of come up again and again in the previous talks, for example, the talk on healthcare that we saw, we're moving away from this idea of training a model to a single task. We're rather kind of training a foundation model that learns all about the data, and then individual tasks are given by specific ways of kind of prompting or conditioning the model. And then just to take that a little bit further, you know, it's often a case of not just conditioning one modality against another, so images against text, for example. You want to do a more kind of fine-grained conditioning where you say, okay, I'm going to specify what some variables are, the ones that I'm conditioning on, but I'm going to leave other variables for you to generate. And so in the case of images, and actually elsewhere, this is known as in-painting, right? So in the examples here, you're saying, well, let's take an image, like the image of the woman's face here, mask out part of the image, and then you're asking the model to generate convincing samples of the masked pixels given the unmasked one. It's still another conditional distribution. So again, in principle, if you have access to the joint distribution over all the pixels, then this is just a kind of a particular sort of joint sample that you're drawing from it. Okay, and in some sense, you know, to take that further, you can say, well, LLMs are kind of like, they're the most flexible conditional models because they're the conditional structure you're always generating from is, you know, just the probability of future text given past text, essentially probability of outputs given inputs. And this is, you know, because language is so flexible, this is where the whole kind of field of prompt engineering comes from, right? You're not changing the underlying distribution of the model, you're just changing the conditioning structure you're feeding to the model. Okay, so all of that is to kind of get across the idea that, you know, we could use generative AI in some sense as a way of telling us, a way of exploring data. So what do I mean by that? Well, in some sense, you know, to learn the joint distribution, you have to learn everything about the data, to learn it properly. You have to learn all the relationships that exist among the many variables in the data. And if you can then access these joint and marginal distributions, you can essentially query the model. You can ask it questions, right? And so I've got a couple of kind of questions that probably look a little bit odd here that you could pose in principle to a generative model. So for example, given this still and the word cat somewhere in the title, what might the rest of the video look like? So the underlying model here is a joint model over videos and captions, let's say. Or you could say, given this image of a person's left eye, how likely is the right eye to be the same color? Now this might look a little bit odd. This isn't how we normally use generative models. We're more likely to say, okay, you know, create a cute picture of a cat for a blog post or something like that. But in principle, you could ask these kinds of very specific questions. And now these look odd in the context of image generation, but they make a lot more sense when you start thinking about scientific data, because in science, we really do want to ask very precise questions like that. And so it's kind of a way of saying, let's think of generative models not so much as, you know, tools or kind of like things that perform specific tasks. Let's think about them as a way of exploring whatever data it was that they were trained on. Okay, so, you know, how does that, in practice, how do we take that and apply it to biological data? So I would say the simple answer to that, my kind of two-word slogan these days is model everything. You know, whenever someone comes to me with a particular data set and says, okay, what, you know, I've gathered all this data, what should I do with it? How can I use AI to help me with this data? I say, okay, take all the data you can get, everything that is related, you know, everything that is kind of interconnected about this domain, and feed it all into one model. Have one model that models the whole thing, right? And then so you're basically saying, try to learn the joint distribution over this entire data set. And now, you know, in biology, it's quite data rich, right? There are large, complex data sets. There's proteomics, there's genomics, there's, you know, large data banks already out there. And, you know, typically in the past, people have taken those data sets and trained specific models for specific tasks. And, you know, I think the new paradigm that's coming is to say, okay, let's not do that. Let's just train a kind of a foundation model on the entire data set. And then think about how, once it's trained, we can access the information in that distribution without having to retrain, right? That's crucial, because, you know, training these models is very expensive and time consuming. If you have to retrain a whole model every time you want to do a different task, this rapidly becomes prohibitive. So kind of like the thing we're looking for here is that we end up with a single model that we can apply to many tasks. And this is maybe even a little bit more kind of extreme than for example, you know, we're used to the idea of kind of multitask learning, but in this case, the tasks are not even kind of known to you at the time when you're training the model, right? You're training the model, and then you're going to ask it, you're going to be able to sort of query any, you know, the dependence of any one variable on any other, or any set of variables on any other set, without having to retrain the model at all. Okay, so to give a concrete example, and the area that you know, I and my colleagues have been looking at is proteomics. And the idea is, you know, you've got something like, you know, the UniProt database, for example. It contains a whole bunch of information, related information about individual protein sequences. So you have, you know, the amino acid sequence itself. You might have a set of, you know, annotations. There's these things called Go terms that are like specific annotations that experts have used to describe the kind of the function of the proteins. There's the three-dimensional structure of proteins. There's the taxonomy, for example, you know, what species was this, a species of animal was this protein found in? And the idea is, well, could we just jointly model this whole thing and then start thinking about, you know, the tasks we could perform just by doing conditional sampling? So, okay, you know, what's one particularly famous task here, one particularly important conditional distribution is the probability of the structure given the sequence, right? This is protein folding. This is exactly what, you know, the AlphaFold model addressed, and, you know, it's now been awarded, you know, the creators of this model have now been awarded 3 Nobel Prizes, right? So it gives a sense of the relative significance here, right? These are, you know, in some sense, they're just probability distributions that we're trying to model like any other, but, you know, it's worth reminding that, you know, these have real sort of like profound implications for science. They're very important. It's about understanding, you know, life could not exist without proteins, as they said in the press release. But then, of course, that's only one joint distribution, right? So if you flip the conditioning structure around and say the probability of sequence given structure, that's inverse protein folding. If you were to try and predict these terms, these annotations, given the sequence and the structure, then you're kind of doing protein function prediction. You're saying, okay, given this protein sequence, what function do I think it performs? And obviously that's a difficult task. If you can generate sequences and structures given some particular conditions, like these binders, then you're kind of going in the direction of de novo design, right? You're actually kind of designing a protein that could be used for a specific purpose, and so on, right? There's lots of other, you know, this is combinatoric here, right? Like all of the different ways you can condition this data. Okay, so obviously that's the hope. Clearly it's not as simple as that, otherwise, you know, we'd already have like a you know, long list of scientific breakthroughs here. There's the usual challenges here that you're going to get, you know, with any real world data, right? You're no longer in the world of sort of nice curated machine learning data sets. It's noisy, it's incomplete, you know, data is collected with different methodologies and so forth. You have the difficulty, and this has come up again and again in the talks we've seen today, you have the difficulty of assessing performance. So, you know, the nice thing about images and text is that you can look at them yourself and kind of judge the performance. You know, the person running the experiments can already get a fairly good handle on what the performance is, and then you can have you can have more systematic ways of, for example, using human feedback to train the model. Those things are difficult in the case of, you know, scientific data, where it's not so easy to define the metrics that you're actually trying to optimize. Another set of difficulties that go along with this type of data is it's, you know, by definition, in some sense, is very heterogeneous, right? If we're talking about gathering together all of the information in some data set, we're no longer kind of, it's not going to look like ImageNet anymore, right? It's not a set of, you know, images that are all kind of roughly the same size with a set of classes from, you know, a predefined, out of a predefined set of classes. It's much more kind of diverse than that, right? It's sequences, tables, graphs, text, images. And I would say this sense of diverse modalities is, I think it's somewhat being addressed by the kind of convergence of machine learning or of neural network architectures, right? Everything is starting to look like a transformer, or at least some sort of attention-based model. So in some sense, that the problem of, you know, how do you model sequences and graphs with the same system, I'd say that's already been addressed in, you know, several of the talks we've looked at. It's often a case of having like maybe a modality-specific encoder, like a CNN for images, like a GNN for graphs, and then getting tokens out of that and feeding them into a transformer. I would say that's probably like the canonical approach there. But there's this last problem, which is the one that I kind of wanted to address most specifically in this talk, is that even irrespective of whether they're drawn from sequences or graphs or whatever, the underlying variables that you're looking at might be discrete, continuous, or discretized. At a minimum, I mean, there are other data classes you consider, like, for example, some things could be cyclical data. But let's say these are probably the three major types. And now the issue there is, well, so, you know, what should you use? If you're trying to model all of this data simultaneously, you're going to end up with a kind of, basically, a maximum likelihood loss function, right? But there's a bunch of different ways of defining that loss function. And I would say the main competitors out there would be, okay, diffusion models, you know, such as are used for image generation, typically autoregressive models like large language models, and then mask prediction, some variant of that, similar to kind of BERT models. And these all kind of have pros and cons. You know, like diffusion models are particularly good for images, they're good for continuous data, and they also allow for very flexible conditioning, right? There's this whole kind of field that's developed around guidance and how to manipulate the sampling process of diffusion models. And that's actually very useful here, because obviously what we're aiming for here is a system where we have a lot of fine-grained control over the conditioning. Autoregressive models are extremely powerful. They're great for discrete sequences. That's what powers the entire large language model revolution. You know, they tend not to be so good for continuous data, although you can apply them to continuous sequences. And they're definitely not good for data where there isn't a natural ordering, right? Like there's a sort of, there's the presumption built in that you're going to model all of these variables in some order. And if you don't know what that order is, like for example, it's an image, it's a graph, it's a video. Well, maybe a video, you could say there's a temporal order at least. But you know, if there isn't an obvious way to order things, it's a little bit problematic applying autoregressive models. And also it's not so easy to conditionally sample, right? Like you typically, your sampling is typically give me the future, you know, sample the future given the past. That's very natural and easy to do with autoregressive models. If you try to do in-painting, you know, if you tried to say, give me the middle word of this sentence given the others, you can do it, but you kind of end up, you know, having to search. Like it's sort of, you're kind of working against the loss function in some sense. And then lastly, BERT-type models. I mean, those are more developed for representation learning rather than actual generative modeling. You can adapt them for generative modeling. Generally, you know, where they've really shone is for representation learning on text sequences or sequences of discrete tokens. But they also, you know, they do have the advantage of there isn't an ordering like there is in autoregressive models. It's sort of built in that it's arbitrary what you're predicting. And because of this mass structure, you do have quite flexible conditioning. Like you can predict any one variable given, or you can predict any set of variables given any set of other variables. So anyway, the final question of all this was, well, we would like to just have a unified system where we use one loss for everything. And we, you know, in principle, you could stitch something together, and probably there are, you know, very powerful multimodal systems out there that do that. But we kind of wanted to say, you know, we're going to be looking at very diverse data sets. We don't want to have to design a separate model, a separate loss function for each one. We'd rather have this sort of unified system. Okay, and the proposal, you know, the system we're working on is called Bayesian Flow Networks. So this was published by myself and colleagues in 2023. So it's relatively recent, you know, kind of approach to generative modeling. And I think the sort of, you know, I'm not going to go into the details. The paper is, you know, long and complex and full of mathematics. I don't want to get, you know, too deep into that in this presentation, but I would say the kind of one-line summary is it's kind of like diffusion models, except you're denoising distribution parameters rather than data. So if we look, and you know, what does that mean? Well, it kind of means that the input to the network is a set of distributions. For every variable in the data, you've got a distribution that represents the kind of current belief about that variable. And the network looks at all of those variables as input, and then it produces a kind of parallel distribution as output. And the key difference is that the output distribution gets to use the context of all the input distributions. So that's where the neural network comes in. The neural network takes in all of these uncertain distributions about individual variables and then kind of integrates over all of them to make a prediction, to sort of re-predict the same variables but with the advantage of context. So if we, you know, how does that play out? So we look at a simple example here of like binarized MNIST. Initially, the system has, you know, it starts off with a uniform prior, so it has 50-50, all the inputs basically just tell it nothing, right? They say that with 50% probability, every one of these pixels is either 1 or 0. That is, it's either black or white. And then as this kind of diffusion-like process, I should say this is sort of, there isn't a forward process here explicitly, like the denoising process of a BFN looks like the reverse process of a diffusion model, for those of you familiar with diffusion models. As the process goes on, the distributions start to shift and they start to become more confident, right? They're starting to say, okay, with some, I've got a certain degree of confidence that this pixel is white and this pixel is black, until you get to, you know, to the end of the denoising process where it's going to exactly tell you with, you know, probability 1 whether everything is white or black. But at every intermediate point along this process, the network is taking all of those noisy inputs and producing a denoised output. So it's essentially, under the hood, it's essentially a denoising model. But really the key point here, and the reason that we think this is a good architecture for, or a good loss function for the types of multimodal data set we're looking at, is that the generative process is continuous even if the data is discrete. And it's continuous because it's working on parameters, right? So that, you know, the parameters of a probability distribution, a discrete probability distribution, are a set of real numbers that can continuously vary. Whereas, of course, the data itself here is discrete, is either 1 or 0. And what's so good about that? What's good about that is basically that you can pass a gradient through it because it's continuous, and I'll kind of expand on that a little bit soon. So this is a kind of animation that we prepared for the paper, which somewhat illustrates, you know, this denoising process. So basically, if you look at this triangle here, this is like the probability simplex for, you know, a distribution over 3 classes. So it's a single variable that can take on one of 3 classes. And basically the belief, you know, the start of the process, all of the probability mass is concentrated in the center. That means it's giving probability 1/3 to all of those classes. The white trajectory shows you how this sort of denoising process progresses over time. And it follows this kind of squiggly random path that ends up in one of the corners. So it goes, you know, ultimately it goes from, you know, knowing nothing about the variable, as in, you know, having a prior that gives uniform probability to all of the classes. It progresses from that to being very confident about what exactly the class is, right? It's whatever the class in the bottom left corner corresponds to. But the key point is that this white trajectory is in fact continuous, right? It's moving in this continuous space, and so we can pass a gradient through it. And yeah, and maybe another way of illustrating the same thing. If we look at I've designed a representation here where we look at the probability given to a piece of text, where we say the size of each letter corresponds to the probability assigned to that letter by the network. This is the plot on the left, the BFN plot. So you can see at the start, you can't really read anything because, you know, all of the letters are small. The network is giving more or less equal probability to all, you know, 26 letters or whatever it is it's looking at. As the process goes on, and this is the output distribution of the network, not the input distribution, but as the process goes on, it starts to become more confident about, you know, predicting certain words, predicting certain letters. It's starting to find patterns in the data. Yes, is there a question? In this case, there's no conditioning. It's unconditional text generation. So it's just generate a sequence. It's just treat text as a sequence of characters and generate a sentence. And this is, I think this is a sentence drawn from the text8 data set. So, you know, really the key takeaway here is just that there's a continuous transformation taking the network from, you know, knowing nothing to having a particular sample about the data. And it's sort of surprising because that's a continuous path through a discrete space. That's the thing that's cool here. And if you contrast that, I think it's probably too small to read, but the path that you get with a conventional discrete diffusion. So diffusion can be applied to discrete data, but the sort of conventional discrete diffusion model basically says, take discrete inputs and randomly perturb them to other inputs. So it's sort of, the denoising process in this case starts off with gobbledygook, this sequence of random letters, and then the letters start to get flipped one at a time with a certain probability until it resolves into something meaningful. And this is also, you know, entirely valid generative process, but because it involves these discrete flips, it isn't continuous and you can't pass a gradient through it. Okay, so this is kind of like, what I've been setting the scene here for is, well, what are we going to want to do with these models? Like I said, the really crucial thing is to be able to perform conditional sampling. And so we've kind of looked at, you know, gone through the literature basically, the sampling methods that we're using for BFNs are all ported from diffusion models because, as I've said, the basic processes are fairly similar. And there's kind of like two main classes of, you know, of sampling algorithm. There's gradient-free ones and gradient-based. And the sort of TLDR here is that the gradient-based methods are better. They're better because they use more information. They use information in the model, telling it, you know, it's kind of like you're saying at each point, I want you to drift the sampling process towards the conditioning information, make the conditioning information more probable. I won't go into too much detail about how these different methods work. You know, the simplest one is you just, when you train the model, you kind of mask certain things out and predict them from other ones. And you know, doing that doesn't require a gradient. This is sort of similar to what's done in classifier-free guidance with, you know, with diffusion models. If anyone's familiar with that, you kind of have a sort of, it's like a dropout on conditioning information. There are particle filtering methods, it's a stochastic Monte Carlo, where you sort of run lots of particles in parallel and sequentially pick the ones that look like they're going to give you the correct conditioning information. Then there is this kind of class of what I would call score-based sampling, where you're explicitly using the gradient. You kind of use Bayes' rule to show you how to get, like, how to follow the gradient using the conditioning information. And then there's like a combination of, you know, methods 2 and 3 in some sense, a combination of particle, the most advanced kind of sampling method that we found is a combination of particle filtering and score-based sampling, which is called twisted particle filtering. And this was published in a paper, I think a 2023 paper on diffusion models. So anyway, there's lots of different ways of, like, maybe the key point to make here is that it isn't easy to do the conditional sampling. So ideal world, we train this one joint model and we do lots of conditional, and then we can just easily access all of these conditional samples. Of course, it's not as simple as that. Conditional sampling is itself hard, and there's a whole kind of field of research here. But at least BFNs are giving us a good framework here because everything is continuous and everything can be modeled with a single loss function. Okay, so just to take a kind of toy example to fix these ideas and explain a little bit more concretely what the process is, we're going to go back to the good old MNIST data set of handwritten images. And in this case, training a joint model means, well, we've got basically two modalities here. We've got images and classes. Classes are very simple. There's just, you know, there's 10 different image classes. So you've got a single discrete variable which has k, you know, 10 different classes, 10 different values. And then the image we're treating as, you know, a set of discretized variables where the discretization gives you the grayscale. So it's basically a number between 0 and 255. And you've got, you know, it's an image. You've got a whatever it is, 28 by 28 grid of those. Anyway, you train the model in this joint distribution. You generate samples. You know, it generates things that basically look like MNIST, right? Like, that's not in itself a big deal, obviously, but, you know, it's obviously like the first sanity check. In fact, in general, for all these systems we look at, the first thing you've got to test is the joint distribution actually right? There's no point trying to sample conditional distributions if you've got the joint wrong anyway. And so you can see here the images look like MNIST images, more or less, and the labels pretty much match the images. So, you know, at least from, you know, eyeballing it seems to be good to go. And then you can say, okay, what are the kind of conditional samples we might want to draw here? Well, one obvious one is to say, let's condition on the class. So if we fix the class to be 1 and then generate the image, we should get a whole bunch of different 1 images, right? And that's what's shown on the left. Likewise, we fix it to 8, we should get a bunch of 8s. That's what's happening on the right. So that seems to be working. And these were done with this kind of fairly straightforward particle filtering approach. This is not a gradient-based approach. You know, it's a relatively simple data set, so it didn't really require that. And then we can go a bit further and say, okay, let's not worry about the class, but let's do a kind of in-painting model. So in this case, we took this image of a 2, shown on the top left, and masked out everything but the top of it, and then said to the network, okay, now generate all of the remaining variables. So it had to generate all of the masked variables, but also to generate the class. And you can see that, you know, what it does is roughly half the time it completes this, you know, these revealed pixels. It completes them as a 2, and roughly half the time it completes them as a 3. And you can, you know, it's easy to see why, right? That with that particular shape, there's only really 2 digits that are gonna fit that pattern. But you know, what's kind of interesting is that, in some sense, we've got a, you know, we're marginalizing over the class distribution, right? We're fitting the we're fitting some of the pixels, some of the image distribution, but allowing the class distribution to be whatever. But then we can go one step further than that and say, okay, let's fix the class distribution as well. So now we're showing it the exact same pixels, but we're fixing the class to 2. And so now when we generate, it only generates 2s, right? Because obviously, otherwise it wouldn't be consistent with the label. Likewise, if we tell it to generate 3s, it only generates 3s. Okay, so this is just the sort of proof of, and sort of one final task is say, okay, let's fix the image and generate the class. And of course, this is just classification, right? So this is just an extremely kind of cumbersome way of training an MNIST classifier, right? Which is like the sort of machine learning 101. But, you know, it works. It gives, you know, 99% accuracy on the test set. It's a reasonable classifier that, but the key point is it wasn't trained as a classifier, right? It was trained as a joint model, and we're just sort of zero shot asking it to operate as a classifier. Okay, so obviously none of that would be interesting if we were only applying it to MNIST. The point is to take that methodology, that framework, and apply it to, you know, really like interesting scientific data. And so our first kind of stab here was what we call prop BFN. This was basically do what seemed like the simplest first thing to kind of prove this method, which was to say, okay, let's just model protein sequences as in amino acid sequences themselves. Essentially just treat them as a sequence of characters. Think of it as if you're just doing, you know, language modeling in an alien language that you don't understand. Or at least that I don't understand it. And I should preface all of this by saying I am absolutely not a biologist, and you know, I apologize for saying naive things about biology. I'm enjoying learning about biology, however. So you know, the first test here was, well, just, you know, can we just model protein sequences full stop, right? And I think the answer here is yes. We've got a state-of-the-art model for you know, unconditionally generating amino acid sequences. It's doing better than diffusion models, better than autoregressive and BERT models on this task. And what, you know, you already have to ask the question, what does better mean here, right? It's not a question of kind of bits per character, although you can measure that. It's really more as a little bit like how you assess image generation, right? You've got these 3 things that you need to have. It needs to generate novel sequences. So you need to know that it's not just copying the training set, otherwise it hasn't learned anything. It needs to generate things that are natural, right? And measuring naturalness is difficult. Like there's a lot of processes there. One of them is to kind of take the sequences you generate, fold them with an existing, you know, pre-trained protein folding model, and then assess the quality of those 3D structures. And then the last one is diversity. So you have to sort of basically make sure there's no mode collapse. Make sure that, you know, as well as generating things that are natural, it's generating things that kind of span the distribution of the training set. And I won't go into the details, but they're all in the paper, which is forthcoming in Nature Communications. Or you can see the preprint on arXiv. But, you know, there was a lot of work went into kind of accurately measuring these metrics to make sure that, you know, the things it's generating were in fact meaningful. But so going kind of beyond that, I'm going to talk you through a more recent development from our group, and this is mostly the work of my colleague Bora. And this is around a kind of, let's say, a subfield of protein modeling, which is antibody modeling. Okay, so this is where my biological limitations will start to show, but, you know, please bear with me. My kind of simplified understanding here of, you know, what an antibody is essentially like this kind of Y-shaped structure at the top left. So it consists of kind of like a pair of identical, you know, chains, each of which contains two kind of sub-pieces, right? So there's the heavy chain and the light chain, and these are the things that are marked with H and L on the diagram. And in particular, we can see this kind of highlighted region where there's a 3D structure kind of showing this highlighted region of the antibody. That's the part that we're really interested in modeling. And even a kind of substructure of that part are these areas known as CDRs, which stands for complementarity determining regions. These are the regions that kind of determine how the antibody is going to bind to antigens, which of course that's sort of like the key property that you want antibodies to have. They have to be able to bind to, you know, diseases. They have to be able to, in order to function. And so then this, you know, this structure can then be broken down further. You have, so the two long sequences at the bottom here show the amino acid sequences for a specific heavy chain and light chain, and these regions highlighted in color, these are the CDR regions. So there's 3 CDR regions in the heavy chain, 3 CDR regions in the light chain. These are like the most kind of interesting things to model. But then there's also a bunch of other things. There's kind of metadata or higher level attributes of the antibody. So things like hydrophobicity and you know, charge imbalance and stuff like that. And these are things that we might want to control, particularly in as much as we're interested in kind of like drug design here, right? Like you need to develop antibodies that have the right properties that you're actually going to be able to manufacture and store them, for example. And there are also the, you know, there are various genetic attributes. So one thing that's sort of shown at the bottom here, you can see there's 3 genes, V gene, D gene, and J gene. They're all controlling they're basically kind of controlling the amino acid sequences in the heavy chain, and there's 2 genes, V gene and J gene, in the light chain. So there's some sort of, we could think of it almost like these are like latent variables here. This is something like the latent structure that determines what the sequences are. Anyway, big picture here, it's complex, right? Like this is just a single antibody. There's a lot of related information. And so we've ended up with actually 45 different modalities. So this, for every single antibody in this observed antibody space, which is a you know, antibody data set, we've got 45 different modes that we're going to try and jointly model, you know, using a single BFN model. And you know, and the idea here, as before, the idea is to kind of train this joint model and then query it using conditional sampling. So, you know, what would these queries look like? They might be something like, given a particular CDRH3 sequence and a particular V gene for the light chain, now generate me a set of antibodies that are human, right? Like where the species attribute corresponds to human. Or you might want to reverse it and say, okay, here's an antibody sequence. Give me all the metadata. Tell me about the hydrophobicity. Tell me about the species. Basically use it as more like a classifier. And the list goes on, right? With 45 modalities. And, you know, it's not just a case of conditioning one modality against another. These can all be kind of in-painted. So you might always be taking a sort of a piece of any one modality. Okay, and so, as I've said, the first thing you have to do with this sort of model is verify that the joint distribution looks good, right? You've got to do some sanity tests here. You know, one of them is to say, okay, what does the distribution of lengths look like for these you know, regions that you're trying to generate, these CDR regions? And you know, this is just a fairly simple marginal distribution, but you can see these plots on the left are showing that, you know, the joint distribution as learned by the model and the underlying distribution match along this. You know, it's impossible to, you know, because we don't have explicit forms for any of these distributions, right? So we can't exactly measure whether we've matched the training distribution. All you can do is sort of like pick slices, pick views of the training data and say, okay, do we match along this dimension? Do we match along that one? And it's basically a case of it's necessary but not sufficient, right? Like no matter how many of these tests you do, you want to make sure they're all right, but you still don't know that you've really got the true distribution. You just don't have an obvious red flag that you got it wrong. And likewise, the T-SNE visualizations, now I forget the exact details here, but those were used to compare, yeah, we're looking at taking the sort of the 3D structure that, you know, we observed when we took the generated sequences and passed them to a folding model and comparing that with the 3D structures in the training set and, you know, measuring these pairwise distances between points, basically. And this, again, gives you a kind of a flat, you know, distribution that at least you can look at, you can analyze fairly straightforwardly. In the end, it's just a case of saying, well, the distribution that we're getting out of our model looks like it's matching the training distribution. Okay, so then the fun begins. And the fun begins when you start doing the conditional sampling. And so in this case, we're using this more advanced sampling method, the twisted stochastic Monte Carlo. And just to kind of, you know, give an exploration of what we might do with this model, well, you might start off with a kind of ancestor antibody. Let's say this is a real antibody from the training set. You start off with that, and then you say, well, what would happen if we removed the green part, basically, which I think is the CDR-H3 region, and we said, okay, now I'm going to fix all the rest of it. So the conditioning information is the stuff kind of shown above the arrow here. That's basically the whole rest of the antibody structure, but I want to change the length of the CDR-H3 region. You know, let's say this one here was, I don't know how long it is, it's 12 residues long or something, and we say, okay, I want a shorter one. I want to generate one that is 5 or 6 residues long. And then, you know, you pick a sample from the model, and that's exactly what it does, right? It gives you back something that is, you know, it's still, you know, all of the conditioning information is left unchanged, but it's had to change this, you know, CDR-H3 region in such a way that it matches your constraints, right? It needs to match with the rest of the structure like it did before, but it also needs to match this new constraint of being shorter. And these are real, this is real data, by the way. Okay, and then you could go a bit further. So once you've done that and you've changed the CDR-H3 region, maybe you want to say, okay, now I actually want to lop off the whole of the light chain. For whatever reason, I'm happy with the heavy chain, I'm not happy with the light chain. So I'm going to keep the heavy chain fixed, and then I'm going to say, okay, I want a new light chain, but I want it to have some particular properties. You're going to specify this V gene that you want the light chain to have, and you're going to specify something like hydrophobicity. And so then again, it's a conditional sample. You run the model forward, and it generates you know, a new chain that, again, has to match all of your constraints, right? It has to match the new conditions you put in, but also the existing structure that was already there. And as I say, these are real samples. And, you know, for me, this was very exciting to see this happen, right? Because obviously BFNs they're an abstract, you know, mathematical model that I developed, and I'd only ever really looked at text and images and sort of standard machine learning data. And then to see it actually work on real data and generate things that look like real antibodies, this was really exciting. And I think that's really like where for me, that's really where the thrill of taking AI to science lies, is like seeing that, you know, these abstract methods can do something, you know, really meaningful with important data. And then, you know, after that, no. Oops. I seem to have a duplicate slide. Okay, so then, you know, just to remind you, this is it's a distribution, right? So like on the previous slide, we said, okay, keep the heavy chain. Basically, give me a sample from the distribution over light chains given heavy chains. Obviously, you can pick lots of samples, right? And here, what we've done is kept the heavy chain fixed and picked lots of light chain samples and just sort of plotted them on top of each other. And that's where you get this kind of spaghetti-like structure. And I think this is a little bit like the GenCast presentation we saw on Tuesday, right? Where you saw like an ensemble of possible trajectories that a tornado could take, for example, or a set of possible kind of atmospheric conditions. It's the same kind of underlying idea here. It's like you're not making a prediction in the classical sense that people used to use, the way people used to think about sort of applying these models to data. You're sampling from a distribution, right? And it's really important that it remains a distribution. It has uncertainty, but the key point, I guess, is that there are lots of different structures here, but all of them are individually plausible. All of them are sharp and distinct, right? There's no such thing as average weather, and there's no such thing as an average protein. There's no such thing as an average face. These models are only interesting in that they pick out kind of diverse and natural samples, and that they give you an ensemble or a distribution over these samples. Okay, so these are nice sort of demos of the model, but just to give maybe a little bit more kind of quantitative support. One task here was to say, well, let's essentially use it as a classifier, in much the same way that we could take that MNIST model and at the end use it to classify images. We can say here, let's use this to classify a whole bunch of attributes about antibodies. So we're talking about specific genes in the heavy and light chain, specific families, this LC locus. I don't know what that is, but it's an important piece of metadata that we might want to predict. We're basically it's gene identification. So we're predicting genes given an amino acid sequence. And the key point here is that compared to Anarchy, which is a kind of like a conventional bioinformatics tool that people have been using for these purposes for a long time, this BFN, this antibody BFN model works much better. And it works much better without having been trained for any of these tasks, right? It's literally just zero shot, sort of change the conditioning and kind of use it as a classifier. And then maybe even more interesting is to say, okay, well, what would we want to use this for? Well, obviously this is kind of about drug discovery, right? And here it's about sort of like guided antibody design. So we're going to say, in this particular case, we've said, well, we've got a particular antibody for a particular disease, or actually, the story I think, is a little bit more complicated than that. Bora looked at the literature for a particular disease and found a whole bunch of antibodies and found what was common among them. So he sort of found that they had a bunch of attributes that seemed to be important. And these are the things that are listed here. These are to do with like chain length and, in some cases it's very specific. It's like saying there is going to be this particular amino acid at this particular location in the chain. And having picked out the attributes that he thought were important, he could then say, okay, I'm going to fix all of those, and now those are my conditioning information, generate me a whole bunch of antibodies. And this thing could generate, lots and lots of antibodies that all kind of match the constraints, but were still natural and kind of plausible. And this is kind of striking, given that there were only 21 of these antibodies, or 21, I think so, 21 of these antibodies that match those constraints in this entire data set of however many million antibodies. So it's kind of like it's able to extrapolate in a really useful way. And you can imagine how useful this would be for someone who's working in drug design. Right now they have so many more candidates that they could look at and that they could analyze in the lab. And yeah, I think that's essentially it. So just to summarize, we have these very powerful generative AI models. I don't think anyone can question that anymore. And what's powerful about them is they can learn these incredibly complex joint distributions. They can generate whole videos. They can generate poems. They can, you know, they can generate songs, whatever you want. If you can take that model and sample from it in a really structured and precise way, then you can ask questions about the data, very detailed and precise questions about the data that it was trained on that you really didn't have access to before, right? So there's a really powerful tool here. And my feeling is that it can become this great exploratory tool for biology, but also for other sciences where there's lots of data. And I think it's key maybe to highlight this term exploratory. Like, so I don't see this as a sort of end-to-end replacement for scientists or even for particular scientific processes. It's much more about enabling people to, or guiding people's search, right? So giving people ideas to look at, right? If they have a particular, you know, in the same way that in the past people would analyze data by looking for correlations between pairs of variables, now they can do something much more powerful and ask much more detailed questions about the data. And I think those can help to guide scientific research. And that's the end of my talk. Thank you.
Thank you for the talk. So actually, I don't have any knowledge on biology, neither on medicine, but I have a question out of curiosity. So my question is about cancer treatment using personalized medicine. And I know that it focuses like on the personal information, like the genes, the lifestyle, and so on. And the goal of it is like to target only the cancer cells instead of all the healthy cells of the body. And this is, let's say, by creating an antibody that recognizes the specific epitope of the diseased cell. So actually my question is AI, is that done using AI, like the target, the creation of the antibody, or the specific selection of the epitope?
I mean, in some ways you are asking the wrong person this question, but I think, I mean, basically it is a process that is, I mean, it isn't ready yet, right? Like, you know, yeah. Personalized medicine for cancer is not yet a reality, but it is certainly something that we're working towards, and I think that AI is going to play a critical role there, exactly in this kind of you know, direction of being able to guide people's search, right? Like, it's going to be certainly, yeah, it's going to be very important to be able to make accurate predictions about you know, the impact of you know, choosing particular medicines for particular people, and how, you know, one person's DNA impacts that, and so forth.
So actually that targets both, like the creation of the antibody and the selection of the epitope, right? That could be applied in both, for both purposes?
I believe so. I believe so.
Okay.
But as I say, I'm afraid I'm really not an expert on
Thank you a lot.
The biological side of this.
Hello. Thank you for your presentation. I have a bit more general question. As far as I see, all machine learning boils down to maximizing log likelihood of the data. So, and when data moves or changes a little bit, then we have to again maximize our model to fit the likelihood. So the term likelihood is the key here. Then there's another school of thought from Judea Pearl, who say, okay, we need to make assumptions on the data generation processes in order for us to be able to do more than just maximizing likelihood. But from what I have seen from them, either there's not much of models that can generalize to different situations. So I'm a little bit puzzled how we are trying to build general artificial intelligence just by trying to maximize likelihood of the data. What's your thought on it, and what do you think? How do we go about making agents learn on their own?
I mean, I think that's quite tangential to this. I mean, this is very much, you know, the method I've presented today is very much about maximizing likelihoods. It's more about learning about the data and being able to explore the data as opposed to kind of creating a model that's really intelligent. But I would say in general that I agree with you in the sense that mostly what we have right now are very powerful models of data as opposed to, you know, truly intelligent models. But I think, yeah, the question of how to get to true AGI, I mean, this is a whole other, so that's a whole other conference, I think.
Thank you. Hi. Thank you for the talk. I think this stuff is really, really cool. I think even 5 years ago, I would not have imagined that we'd be at this stage. My question is, I think this is a really cool application of this sort of model. Is there any other domain that you'd be excited to try it on?
Yeah, lots. I think honestly, it's more a question of frustration at how long it takes to work on any given domain. I mean, you know, already sitting alongside proteomics, there's genomics, right? And we'd like to be looking at DNA sequences and RNA, and you know, but there's only so many things you can look at, and there, you know, it's never as simple as, you know, you just come to this new field and you shovel in the data and you get the result, right? The devil is always in the details. I think it's kind of like, it's limitless. Like I say, like to me, the ideal is something like a method where you literally can take, you know, pretty much any like empirical data set, and someone who isn't even a specialist, who doesn't even understand the model, could just feed it into the machine and start asking it questions. That seems to me like the ideal. Maybe they have to annotate the data correctly and you know, give a little bit of information about, you know, how it was collected and so forth. But that's still some way off, and it feels like there's always a lot more work still than there should be. Like every new data type, every new data source still involves too much manual labor at the moment. That's kind of what's getting in the way. You know, the big successes of generative AI are in those modalities where, like text and images, where you can get an enormous amount of data that's all fairly homogeneous. You know, you don't have to worry so much about how to, or let's say at least the mechanisms that you can use to present that information to the network are already understood. Yeah, so I think the answer is there's too many to count. Anywhere where you've got really complex relationships in data, which is pretty much all, you know, interesting scientific data, I think this type of approach, I mean, weather forecasting, you know, as we saw a couple days ago, and healthcare today, these are both areas that are kind of, you know, it's a similar approach, right? It's like this idea of just like model the data with a generative model and then, you know, draw samples, basically.
Okay, we can do one last question. There's a microphone that, oh, yeah.
Hello, thank you for the presentation. It was really good. I want to ask to have insights about the accuracy of the models, because it's related to biology, to science, and also you talked about heterogeneous data. So did you try different combinations, or there is a certain combination of data you tried?
So yeah, maybe the first question, accuracy, like this is really important, and this sort of goes back to this idea of evaluation and how it's difficult. You know, like every scientific field, even just like every subtask, there's going to be a different metric, basically, and you know, you have to kind of obey that metric, right? Like it has to be, like for example, if you're doing protein folding, you know, people measure, for example, like the RMSD between the ground truth and the structure you generated. There's, you know, it's basically you know, you can't really, a model that hallucinates is sort of just about okay for you know, a chatbot or for an image generator, but scientifically speaking, you don't want that, right? You want something that is much more precise. So yeah, you have to do a lot of careful calibration and a lot of work on evaluation there. The second question, no, can you remind me what that was?
Yeah, I asked, you used heterogeneous data, so did you try many combination of data, or it was a certain combination you used?
Oh yeah, so I mean, you know, the ideal was always, let's just take all the data and model it all at once. In practice, it's often a case of sort of 1 to n, you know, like you start off with one of the modalities, check you can model that, then you throw in another one. If that looks like it's looking good, then maybe you can throw in all the rest. But that's pretty ad hoc, I would say. I think in general, it's not that if the model is learning the joint distribution well, it's not going to be all that sort of fragile to, you know, like if you removed one modality, it wouldn't suddenly break, basically. You know, it often just comes down to sort of how much time you've got to process the data and how much you can fit in memory at once. It comes down to quite practical considerations often. Yes.
Thank you so much, Alex. Did you have anything else?
There was one more question.